Bioprinting is an incredible technology that sounds like it comes straight from the pages of a science fiction novel, but it's very real and has the potential to revolutionize medicine as we know it. At its core, bioprinting is like regular 3D printing, but instead of using plastic or metal, it uses living cells and special bioinks to create three-dimensional structures. These structures can be anything from simple tissues to complex organs like hearts and kidneys. Now, why is this such a big deal for medicine? Well, think about it this way. If someone needs a new organ, like a liver or a lung, they usually have to wait for a donor, and that can take a long time. But with bioprinting, we could potentially print these organs using a patient's own cells. This means no more waiting lists, no risk of rejection because it's made from your own cells, and ultimately, it could save countless lives. It's like a medical superhero that has the power to create new body parts when we need them the most. Let's delve into the fascinating world of bioprinting and explore these three cutting-edge technologies: inkjet-based, laser-based, and microfluidics bioprinting. Inkjet-based bioprinting mimics the mechanics of traditional inkjet printers, but with a remarkable twist. Instead of text or images, it crafts living tissues. This technique employs specialized bioink cartridges filled with living cell suspensions. These bioinks are meticulously deposited layer by layer onto a substrate, akin to constructing a tissue lasagna, one cell layer at a time. Laser-based bioprinting takes precision to a whole new level. Utilizing lasers, it selectively positions cells into a substrate. Laser pulses gently propel tiny droplets of bioink into the desired locations. This high-precision method provides intricate control over cell placement, enabling the creation of complex tissue structures with meticulous detail. Microfluidics bioprinting operates on a fundamentally distinct principle, employing microchannels and fluid dynamics to govern the flow of bioink. Cells suspended in a liquid medium are directed through microchannels to precise locations where they form tissue structures, akin to a miniature biological conveyor belt orchestrating the assembly of tissues at a microscopic scale. These technologies share a common objective: creating functional biological tissues. The journey begins with the formation of bioinks, meticulously prepared mixtures of chemicals, biomaterials, and growth factors tailored to the specific bioprinting method. Inkjet-based bioprinting relies on thermal or piezoelectric actuators, expelling minuscule droplets of bioink into a substrate to construct tissue structures layer by layer. In contrast, laser-based bioprinting leverages lasers to generate pressure pulses that propel cell-laden droplets onto the substrate, ensuring precise control over cell placement. Microfluidics bioprinting ingeniously utilizes microchannels to manipulate fluid flow, guiding cells to designated locations where they are deposited to form tissue structures. Each bioprinting technology boasts distinct strengths and limitations, rendering them suitable for diverse applications and research objectives. Inkjet-based bioprinting offers speed and scalability, while laser-based bioprinting excels in precision. Microfluidics shines in creating microscale structures. As bioprinting relentlessly advances, these technologies will assume pivotal roles in the transformative fields of regenerative medicine, drug testing, and tissue engineering. Whether through inkjet, laser, or microfluidics, bioprinting propels us on a captivating journey through the realm of science fiction to reality, where the future holds astonishing possibilities. Taking a deeper look into the bioinks that make bioprinting possible requires us to understand the roles played by cells, biomaterials, and growth factors on the human body. Cells are the building blocks of life, and they are the heart and soul of bioinks. In bioprinting, cells can be derived from various sources, including stem cells, primary cells, or cell lines. These cells are selected based on the tissue or organ being printed and must be compatible with the intended application. The significance of cells in bioinks lie in their ability to self-organize and differentiate into specialized cell types. For instance, in the case of printing heart tissue, cardiac cells are used. These cells will form functional heart tissue over time, making the printed structure biologically relevant. Biomaterials act as the support structure for cells within bioinks. They provide the necessary mechanical stability and structural integrity to the printed tissue. The choice of biomaterials is crucial, as they need to mimic the extracellular matrix, which is the natural scaffold that surrounds cells in the body. Common biomaterials used in bioinks include hydrogels, like alginate or gelatin, and synthetic polymers, such as polyethylene glycol. The biomaterials should be biocompatible, biodegradable, and have the right physical and chemical properties to support cell growth, adhesion, and tissue development. Growth factors are like the conductors of the tissue-building orchestra. They are signaling molecules that regulate cell behavior, including processes like cell proliferation, differentiation, and tissue development. Growth factors are often added to bioinks to provide cues to the printed cells, directing them to follow specific developmental pathways. For example, in bone tissue engineering, growth factors like bone morphogenetic proteins are used to promote bone cell differentiation and mineralization. The significance of these components within bioinks cannot be overstated. They work in synergy to create a microenvironment conducive to tissue growth and maturation. The cells bring the biological functionality, the biomaterials provide the structural framework, and the growth factors orchestrate the entire process. Together, they enable the formation of complex functional tissues through bioprinting. Moreover, the precise formulation of bioinks is tailored to the specific requirements of each bioprinting technique. Inkjet-based bioprinters, for instance, demand a bioink with the right viscosity to be ejected in droplets. Laser-based bioprinting relies on bioinks with suitable optical properties to interact with lasers effectively. Microfluidics bioprinting necessitates a bioink that can flow smoothly through microchannels. In essence, the composition of bioinks is a delicate balance, and researchers continually fine-tune it to optimize bioprinting processes. As bioprinting technology advances, so does our ability to create functional tissues and organs, promising remarkable breakthroughs in regenerative medicine, disease modeling, and personalized medicine. Perhaps one of the most astounding achievements in bioprinting is the successful fabrication of functional human organs within laboratory settings. Researchers have achieved this by meticulously replicating the intricate vascular networks and cellular arrangements necessary for organ functionality. This monumental accomplishment promises to address the perpetual shortage of donor organs available for transplantation. Moreover, the use of a patient's own cells in the bioprinting process substantially reduces the risk of organ rejection, marking a groundbreaking advancement in regenerative medicine. Bioprinting has emerged as a potent catalyst for advancing skin and tissue regeneration. Scientists have pioneered techniques enabling the printing of living skin tissue, complete with multiple cell types and a fully developed vascular network. This development carries transformative implications, particularly for individuals suffering from burn injuries, chronic wounds, or those in need of skin grafts. Bioprinted skin and tissues facilitate swifter healing and contribute significantly to minimizing scarring, promising improved quality of life for patients. Another paradigm-shifting application of bioprinting lies in drug testing and disease modeling. Researchers can now construct intricate 3D tissue models that faithfully replicate the physiological intricacies of human organs. These models serve as invaluable tools for assessing the safety and efficacy of new drugs, substantially reducing the reliance on traditional animal testing and expediting the drug development pipeline. Furthermore, bioprinted tissue models offer a highly realistic context for the study of diseases, providing profound insights into disease mechanisms and potential therapeutic interventions. These remarkable breakthroughs underscore the transformative potential of bioprinting across a spectrum of fields, from regenerative medicine to pharmaceuticals. As bioprinting technology continues its rapid evolution, we anticipate even more remarkable developments on the horizon. These advancements bring us ever closer to the era of personalized medicine and regenerative therapies, fundamentally shaping both healthcare and our comprehension of biology. Bioprinting is poised to revolutionize the healthcare industry by offering unprecedented opportunities for innovation and improvement. At the heart of this revolution is the concept of personalized medicine. With bioprinting, it becomes possible to create tissues and organs using a patient's own cells. This not only reduces the risk of transplant rejection, but also eliminates the need for immunosuppressive drugs. Patients could receive organ replacements tailored to their unique anatomical and genetic characteristics, enhancing the safety and success of medical treatments. Additionally, bioprinting is reshaping the landscape of drug testing and development. Traditional methods often fall short in accurately predicting drug effects on the human body. However, bioprinted tissues and organs offer a more realistic platform for drug testing, leading to safer and more effective medications. Pharmaceutical companies can assess drug safety and efficacy with greater precision, ultimately expediting the drug development process and benefiting patients with faster access to new treatments. Beyond drug development, bioprinting also plays a pivotal role in disease modeling. By creating 3D tissue models that closely mimic human physiology, researchers gain deeper insights into diseases such as cancer, Alzheimer's, and heart disease. This advanced understanding facilitates the development of targeted therapies and reduces the reliance on animal testing in preclinical research, contributing to more ethical and scientifically rigorous studies. Moreover, bioprinting's applications go beyond treatment to include rehabilitation and prosthetics. Custom prosthetics and orthopedic implants created through bioprinting can enhance patient comfort and functionality. This innovation improves rehabilitation outcomes and enhances the quality of life for individuals with limb loss or orthopedic issues. In the realm of surgical planning and training, bioprinting facilitates the development of lifelike surgical models. Surgeons can practice complex procedures on these models, refining their skills and improving surgical precision. This reduces the risk of complications during actual surgeries and enhances patient safety. Ultimately, the transformative potential of bioprinting extends to reducing overall healthcare costs. While there are initial investments in developing and implementing bioprinting technology, the long-term benefits are significant. More efficient drug development, reduced organ transplant waiting times, improved treatment outcomes, and streamlined healthcare processes can lead to substantial cost savings within the healthcare system. Bioprinting is reshaping the healthcare landscape by advancing the fields of regenerative medicine, drug development, disease modeling, and personalized treatment. Its potential to address some of the most pressing challenges in healthcare promises to improve patient outcomes, reduce costs, and drive innovation in medical science and practice. As this technology continues to advance, its transformative impact on healthcare is expected to grow even more profound in the years to come. Bioprinting, with its promise of creating functional human tissues and organs, faces several interrelated challenges that must be tackled to fully realize its potential. Among these challenges, tissue vascularization stands as a pivotal hurdle. Without a proper vascular network, bioprinted tissues struggle to sustain themselves, receive vital nutrients, and eliminate waste efficiently. Achieving the intricate architecture of blood vessels within bioprinted structures is essential for their survival and functionality. Simultaneously, maintaining cell viability is another paramount challenge in bioprinting. The printing process itself can subject cells to various stresses, from mechanical forces to temperature fluctuations. Ensuring that a significant portion of the printed cells remain viable is crucial for the success of bioprinted tissues. High cell viability not only contributes to the overall functionality of the tissue, but also determines its long-term success within the recipient's body. To address these challenges effectively, standardization becomes a critical consideration in bioprinting. The lack of standardized protocols and quality control measures can hinder progress in this field. Standardization efforts are essential for ensuring that bioprinted tissues consistently meet the necessary criteria for functionality, safety, and reliability. This encompasses everything from formulation of bioinks to precise printing parameters and the characterization of cells used in the process. Moreover, the issue of immunocompatibility looms large in bioprinting. Bioprinted tissues and organs intended for transplantation must seamlessly integrate with the recipient's immune system to avoid rejection. Achieving immunocompatibility involves not only the careful selection of cell sources, but also the development of strategies to modulate immune responses to the bioprinted structures. It's a multifaceted challenge that needs to be addressed comprehensively to ensure the long-term success of bioprinted transplants. These challenges in bioprinting are interconnected and pivotal for the field's advancement. The need for tissue vascularization and the maintenance of cell viability are intertwined with the goal of creating functional bioprinted tissues. Standardization efforts play a central role in ensuring consistency and reliability across bioprinting processes, while addressing immunocompatibility is essential to guarantee the long-term viability of bioprinted organs and tissues within the human body. Tackling these challenges collectively is essential to unlock the full potential of bioprinting in revolutionizing regenerative medicine and healthcare. Bioprinting, an innovative and rapidly evolving field at the intersection of biology, engineering, and medicine, holds the promise to reshape the landscape of healthcare in remarkable ways. By combining the principles of 3D printing with the complexities of living cells, bioprinting enables the fabrication of intricate human tissues and organs. This breakthrough technology addresses critical healthcare challenges, from organ transplantation shortages to personalized drug testing and disease modeling. Bioprinting has already demonstrated its potential by creating functional tissues, advancing drug development, and accelerating research into diseases. As the field matures, bioprinting stands poised to not only transform patient care and outcomes, but also to fuel new avenues of medical discovery and innovation. The impact of bioprinting on healthcare is poised to be transformative. With the ability to produce personalized tissues and organs, bioprinting offers a solution to the scarcity of donor organs and the complexities of immune rejection. Patients could receive tailor-made transplants, reducing wait times and increasing the likelihood of successful outcomes. Beyond transplantation, bioprinting's applications in drug testing provide a more accurate representation of human physiology, leading to safer and more effective medications. Disease modeling with bioprinted tissues offers insights into diseases' mechanisms and the development of targeted therapies. As bioprinting technology matures, its integration into clinical practice has the potential to usher in a new era of personalized medicine, revolutionizing healthcare by offering tailored treatments, reducing healthcare costs, and improving patient outcomes. As always, thank you for taking the time to learn with me. If I missed anything or got anything wrong, please let me know, as I would love to learn more. If you liked what you heard, please consider subscribing. I appreciate the time you have spent watching this video. No matter who you are, you only get 24 hours in a day. Thank you for spending a portion of that with me. See you next time.